RNA, which consists of nucleotides, exhibits living characteristics. It is able to carry out metabolic functions, growth, replication, etc. Autocatalysis. The ability to perform functions for survival, self-sustaining. Replication. When water cools, free-floating/loose nucleotides base pair attach with bases on the RNA strand. The RNA strand then acts as a template strand. Purine nucleotides bind with pyrimidines. Cytosine pairs with guanine. Uracil pairs with adenine. Using hydrogen bonds. Hydrogen atom electrostatic attraction to nitrogen atom and oxygen atom, which is complementary/matches. The sugar-phosphate backbones of the newly attached nucleotides form bonds, phosphate group to ribose, forming a phosphodiester bond. Then when the water becomes heated, the hydrogen bonds between bases break, resulting in two identical RNA strands. This is called semi-conservative replication. Genetic continuity. New strand inherits/has the same sequence of nucleotides, genetic information. The new complementary strand can then also act as a template strand for new complementary strands to be formed. A self-replicating RNA molecule has never been observed in nature, but a theory is nature, for example motion of water, causes it to fold up on itself and duplication occurs. Asexual reproduction is reproduction independent of a partner. Other factors can also promote RNA interaction, such as metal ions which have a positive charge can bind to RNA and neutralize RNA's negative charge, which can stabilize the RNA structure and reduce the likelihood of RNAs repelling each other, as well as acting as a scaffold to accumulate RNAs in closer proximity and stabilize interaction. Minerals contained in clay can also act as a surface for RNAs to bind and accumulate, as well as can offer a shielding effect and preserve them from environmental factors that can disrupt or prevent interaction. And UV radiation, which can excite electrons in reactive molecules, which then participate by providing energy necessary for bonding. Cells would later attain the ability to self-sufficiently synthesize nucleotides. Purine synthesis. Mainly synthesized via salvage pathway, which is where nucleotides are synthesized from products of other pathways and involves 10 reactions using 6 enzymes. Pyrimidine synthesis occurs mainly via the de novo pathway. De novo means anew, and so the nucleotides are made from scratch using simpler molecules such as carbon dioxide, amino acids, and sugars using 6 reactions using 6 enzymes. RNA evolved into DNA, deoxyribonucleic acid. DNA uses deoxyribose, a pentose sugar, instead of ribose. Loss of an oxygen atom from a hydroxyl group, which is one oxygen atom bonded to one hydrogen atom, on the ribose, called oxidization, and therefore it is no longer a hydroxyl group / the loss of the hydroxyl group, which forms deoxyribose. This makes DNA less reactive with other molecules, which increases its stability. Absence of the hydroxyl group creates higher affinity for thymine, a nitrogenous base, instead of uracil. Thymine is also a pyrimidine. The addition of a methyl group, which is one carbon atom bonded to 3 hydrogen atoms, to uracil forms thymine. This enhances the specificity of the hydrogen bonds with adenine, which makes DNA more stable. Also, the absence of the hydroxyl group decreases flexibility and instead provides rigidity, so that now one strand is able to base pair with another strand more efficiently. Therefore becoming double-stranded, called duplex, which is more stable so they can form longer chains, therefore storing more genetic information. Human RNA strand consists of up to 3,700 nucleotides. DNA strand consists of up to 250 million nucleotides. 2 chains of polynucleotides / 2 DNA strands, when joined together coils / twists into a helix because of factors such as the electrostatic repulsion between the phosphate groups in the backbone cause the strands to twist in attempt to adopt the most stable configuration. Non-uniformity in size and shape of the nucleotide base pairs. and the phosphate deoxyribose alternating pattern in the backbone contributes to twisting. DNA is wider at the middle where the base pairs are located and narrower at the outer backbone, called DNA helix roll. The twisting allows the DNA to have a more compact and stable structure, has a shielding effect for the bases to protect them from external factors, and increases alignment for the bases to bond. This forms the double helix, a DNA molecule. One end of a DNA strand, also same on an RNA strand, is 5', written as 5', and ends with a phosphate group. The other end is 3', written as 3', and ends with a hydroxyl group. When two DNA strands bind to form a DNA molecule, they are antiparallel, which means two strands go in opposite directions. 5' next to 3', 3' next to 5'. In eukaryotes, DNA molecules become tightly coiled around 8 histones, proteins, histone octamer. A nucleotide's phosphate groups, which are negatively charged, are electrostatically attracted to histones, which are positively charged, which forms hydrogen bonds. The DNA histone complex is called a nucleosome. There are 1.3 million nucleosomes in humans, and are held together by linker DNA, which forms one long DNA molecule, called chromatin. Chromatin can be organized in various ways, which is mentioned in the next chapter/video. When chromatin becomes condensed together during cell division, they are then called chromosomes. Chromosomes occupy their own certain territories within the nucleus due to factors such as nuclear structures which can act as anchors or scaffolds. Lamina-associated domains on the DNA which allow binding to the nuclear lamina, which is a network of proteins lining the nucleus inner membrane. And interactions between chromosomes. The structure of DNA was discovered in 1953 by Francis Crick, James Watson, and Rosalind Franklin, and were awarded the 1962 Nobel Prize in Medicine. A human cell contains 46 chromosomes. 23 pairs, 23 from mother, called maternal, and 23 from father, called paternal. One chromosome from mother pairs with one chromosome from father. Homologous. 22 pairs are autosomes. The 23rd pair are the sex chromosomes, which determines gender. An X plus Y pair equals male/masculine traits, and an X plus X pair equals female/feminine traits. When cells multiply, every new cell inherits identical 46, 23 pairs of chromosomes. If DNA was unraveled from one cell, it would be 2 meters long. 80% of cells are red blood cells which don't contain DNA. If all of the DNA in a human was lined up, it would reach from Earth around the Sun and back again around 40 times. Organism with the most chromosomes is the Ophioglossum, adder's tongue fern, which contains 1,400. Chromosomes contain genes which are located on a locus, plural loci. The study of genes is called genetics. Genes are a sequence of nucleotides which are the blueprints to create an organism. Make proteins and RNA. Transcription next chapter/video. All genes in an organism/cell is the genome. The study of genomes is called genomics. A human cell contains 20,000 to 25,000 genes. Organism with most genes is the Daphnia pulex, water flea, which contains 31,000. A gene consists of up to over 2 million base pairs, BP, nucleotides. A kilobase is 1,000 base pairs, a megabase is 1 million base pairs, and a gigabase is 1 billion base pairs. All of the genes in a cell which makes up the entire human genome consists of 3.2 billion base pairs. Organism with the largest genome is Amoeba dubia, which has 670 billion base pairs. Genes consist of codons. A codon is 3 nucleotides in a row, and makes up the genetic code. Each codon represents /signals for a single amino acid. For example, UAU or UAC represents tyrosine, UUU or UUC represents phenylalanine, etc. The 4 nitrogenous bases can create/be arranged into 64 possible configurations, 64 codons. Amino acids from the same families are represented by similar codons on genes. Every organism is made of the magic 20 amino acids. More than 100 exist in nature, only 20 are used for protein synthesis, called proteinogenic amino acids. Amino acids are an alphabet and proteins are words. 12 or more amino acids bonded is a polypeptide. Genes are segmented into introns, non-coding, and exons, coding. Intron-exon, intron-exon. Only 2% of genes code for proteins, 98% are non-coding, called junk DNA. An exome is the part of the genome that consists of exons. Genes contain alleles. The genome is 99.9% the same among humans. Alleles are what create diversity. Humans have 2 alleles on each gene which are called diploid, which means the chromosomes are in pairs, one from mother and one from father. With 2 alleles, and if, for example, the first allele inherited from the father codes for brown eyes, and the other allele inherited from the mother codes for blue eyes, if brown eyes are expressed, that is the dominant allele/trait. And blue eyes, which is the allele not expressed, is the recessive allele/trait. The dominant allele is always the allele that is expressed. Which allele is dominant or recessive can be random, or there can be higher probabilities that a particular allele will be dominant. Mendel's third law. Law of dominance. Discovered in 1865 by Gregor Mendel, the father of genetics. If both alleles on a gene are identical, they are called homozygous. If different, one dominant and the other recessive, they are called heterozygous. All alleles in an organism is called an organism's genotype. The express traits that the genes code for, proteins and RNA, is called an organism's phenotype. Morphology is the study of external and internal form and structure of organisms. Functional morphology is the study of organism function. A proteome is the full range of proteins a cell can produce. Humans contain up to 400,000 different proteins, and up to 200 million different proteins exist in the world. Proteins carry out the processes and maintain the function of a cell. Narration derived from Direct Biology, written by hnorsworthy, which is available to buy on Amazon. Thank you to Copenhagen City Explore for giving permission to use the Copenhagen cycling videos in the background.